So the cell communication, okay? Let's take a look at that question, the chapter. Okay, so cell communication, I think we talked about a little bit. So cell communication is where one cell try to relay a message to the other cell, okay? So based on our discussion, it could be a long distance messaging, it could be a short distance messaging, so it could be something we call paracrine, or it could be endocrine. Right? So I mean, again, there's a nerve stuff also as well, but that's a whole different chapter. Okay. So the pancreas is something like if the cell is like nearby, that's a short-term messaging, and long endocrine is well long-term, not long-term, sorry, long distance and short distance. Okay. Endocrine guys, by the way, is what? Yeah, dude, it's like really far away. So this is where it produces. So this guy produces some sort of a signaling molecule, and then send it off to the other cell by the bloodstream. Okay, and that's basically what we call the endocrine system in our body, right? So, so basically the question is not how they produce these hormones or the signaling molecules, right? They call these ligands in this chapter. Okay. That's not the topic for today, because you should already know that how they produce it. So in production, it depends on, so for example, guys, a lot of times they talk about, testosterone. Okay. Sometimes they talk about, guys, I mean, the most talked about is like ADH, okay? Anti-diuretic hormones, okay? Sometimes they call this vasopressin. Insulin is something they talk about also as well, okay? They also talk about something we call, guys, adrenaline, okay? Or epinephrine, okay? Whatever, how you're going to call this. So they take a look at these things, okay? They take a look at it, guys, okay? So the production of these by the cell for this who wants to send the message is not, like, production is not a problem right now, okay? So I think we talked about it, guys, insulin, for example, guys, or epinephrine, okay? These are what we call, what's called as, guys, peptide hormones, okay? It's called peptide hormones, guys. So peptide hormone is what, guys? Yeah, this is these are like proteins, right? So it follows a channel of like, the transcription, translation, in the ribosome, and then it gets shipped out by the cell, and they're gonna put that into a bloodstream, and what? It follows the blood and what goes to the target cell. Okay, so that's what insulin and adrenaline is, or epinephrine. Okay, but now like testosterone, for example, okay, this is actually a steroid, okay, which is steroid. This is called steroid hormones, okay, by the way. So they're smaller as well, okay, and these are lipids, by the way, okay, lipid hormones. Some of them are tinier, but these are now lipids. So now question becomes, okay, so yeah, the question is not how it gets there, okay, that's a different chapter. That's the circulatory system chapter. But now, how will the recipient cell, okay, respond to this? Okay, that's what this chapter is about, okay, how are they gonna respond to it? So keep in mind, guys, one thing I can tell you is this, okay? The reason I'm like squeezing in like cell communication and cell cycle together is there are not that many questions on this for the Olympiad, by the way. Okay, but I technically don't really have to do this chapter at all, because I mean they have a very minimal questions on this one. Okay, but I gotta do it. I have to do it anyways. For this is a base. The other chapters use this as a baseline as well, so I can't just not just pass through this and just go something else. And also, what else, guys? Your AP also has these as well, so I can't just like bypass the chapter. But you'll see like very small number of chapters this will be on the open exam, by the way. Okay. So, but yeah, so I'm not gonna go like too crazy in detail, okay, for this chapter. Okay. So one of the things is like, if a lot of times the questions to given with these two, okay, insulin or epinephrine or adrenaline, they're the same thing. Okay. So if you look at this, so one of the most obvious ones you're gonna get is what we call G protein coupled receptor, okay? G protein coupled receptor. I think we talked about this a little bit last week, right? So dude, like, nothing different here, okay? So we have a receptor protein here, okay, receptor protein for G protein coupled receptors. So these are called the receptors for the cells. Okay, receptor. And now insulin molecules, insulin, right? These guys are peptide hormones, peptide, okay? Which means that these guys are hydrophilic. These guys are hydrophilic. They will not be able to come into the cell, okay? They cannot just pass the membrane because what? They're hydrophilic, okay? So which means they have to rely on a receptor that's on the cell membrane, okay? They cannot come in, okay? They have to rely on the cell membrane receptors, okay? So, but the thing is, I mean, this is what they call G protein coupled receptor. Okay, so they love the term coupled. Okay, so why? I have no clue. Okay, so this receptor is what, guys? I mean, yeah, this receptor has an enzyme, okay, which is a G protein. Okay, coupled with the receptor. Okay, so yeah. So basically, they call this a G protein because it has either GDP or GTP on them. Okay, like like guanosine, diphosphate, triphosphate. So basically, what will happen is, guys. Okay, so we talked about it a little bit, guys. When this they call this ligand. Okay, when ligand binds to the receptor. Okay. Then, I mean, dude, like we talked about it, guys, but it's just so you know, like this is like a substrate, okay, and the enzyme that will, like break this down. I mean, so ligand doesn't really get broken down like that, but just so you know, but if they bind to, what happens, guys? Yeah, like they will react, by hydrogen bonding and stuff like that, like van der Waals force of attractions and stuff like that, and which will change the shape or the configuration of the enzyme overall, to change the shape of the receptor protein overall, which will change what? The G protein shape as well. So what I'm saying is if the receptor gets activated, okay, it will activate the G protein as well. And then once G protein is activated, what happens then? The GDP will get swapped out with the GTP. Okay, GTP is what? It's more active form, right? Triphosphate is more active than GDP, which is diphosphate, right? So once the G protein is activated, what happens? We talked about it, right? What will happen is this will what? Try to produce or try to activate an enzyme that will produce what we call a secondary messenger. Okay, so because the primary messenger, which is a ligand, they will not be able to come in. So which means what? They have to produce a secondary messenger. They'll relay the message down to the cell, right? That's called a secondary messenger. So the secondary messenger, guys, the most obvious one that we talked about is what? Cyclic AMP. CAMP or whatever that your teacher wants to call it, guys. It's called cyclic AMP, okay? Sometimes the different ones, guys, okay? IP3. There's a GAP. So there's one more, I forgot. So I gotta look back at my notes, guys. Glyceracyl something. I forgot the last term here. And then also calcium 2 plus. These are the ones you have to know. Olympiad, guys, these are the ones you have to know. Okay? Get my notes. So IP3, DAG. I'm sorry, guys. DAG. It's DAG. What I wrote here. Diacylglycerol. Oh my bad, guys. DAC. DAG. Diacylglycerol. Okay. So these are the only the secondary messenger that's known or that you guys have to know. Okay, for even for the Olympiad. Okay, like for you guys, the AP Bio guys, just this. That's it. Okay, cycling AMP is the only one you have to know for the AP Bio. But for other stuff, guys, you have to know all three. So it's in the reading, by the way, okay? So they will tell you which one it is and what it is and stuff like that, okay? So one thing to note, okay, that we talked about last week is what? Production of this one. At least this one, okay? So there's an enzyme that we call adenylyl cyclase, okay? Adenylyl cyclase, okay? This is the enzyme that what, guys, will convert the ATP, okay, we'll do ATP, will turn into cyclic AMP, okay, and they'll throw out 2 phosphates. Okay, pyrophosphate. So that's the enzyme, okay? The G protein will activate adenylyl cyclase, okay, and then you produce cyclic AMP, okay, which is what they consider as a secondary messenger, okay. And now, depends on what kind of message it is, what kind of a G protein it is, what kind of cell it is also as well. Okay, they can produce what? Either these two as well. Okay, IP3 or DAG. Okay, calcium 2 plus. It itself being an ion. Okay, like they don't produce calcium 2 plus is an ion. It's not like they can produce it. Okay. But I can explain how they can use calcium 2 plus here, okay? But now, as we know, guys, secondary messenger cyclic AMP, okay, can be, produced, and since this is the one that they ask the most, so this guy is what, guys? So up until here, up until production of secondary messenger, this portion is what they call the reception, okay? It's called the reception, a portion of the signal, cell communication, okay? Keep in mind, there are different receptor proteins. Not only this, they got different ones there as well. They have a tyrosine kinase receptors, okay? They have a ligand ion gated channels. Those are really obvious ones. So tyrosine kinase receptors. I don't think I want to talk. I don't think I'm going to talk about it today. So because of the timeline that we have, guys. So, but again, we'll discuss that after we talk about GPCR. Okay. So, but now, what will happen is, okay. So if in case, guys, the secondary messenger was produced will be, for example, guys, IP3. So if they produce this, okay, IP3 instead. So keep in mind, guys, IP3, okay. So basically, a lot of times, guys, okay, what this guy will do as a secondary messenger is it opens up a reservoir for calcium 2 plus. Okay, so what I'm saying is, okay, there is a reservoir for calcium 2 plus. So from the first few chapters that we talked about, okay, yeah, which organelle stores calcium 2 plus? Do you remember, guys? Do you guys recall, guys? Yeah, which organelle stores calcium 2 plus? What do you think? Vacuole? No. Anyone? Yeah, what do you think?
Multiple choice.
The nucleus, mitochondria, chloroplast, okay, Golgi apparatus, okay, smooth ER, rough ER, I don't know, you tell me, lysosome, okay, peroxisome, glycosome, glycoxome, and then there are many other, not many, but those are the ones that you should know about the organelles, right? But my question is, the vacuole is too general, by the way. That's but which of them, one of the functions for them is storage of calcium 2 plus ion. You're not wrong, by the way. Okay, mitochondria does have some calcium 2 plus in the matrix, by the way, but storage of calcium 2 plus occurs in the endoplasmic reticulum, which is ER. Okay, smooth ER also, for example, they have a calcium 2 plus reservoir. Okay, they stored calcium 2 plus ions. So they store them, calcium 2 plus, and then I'm not kidding, man. Okay, so basically what happens is this IP3. Okay, keep in mind, guys, the intracellular concentration of calcium 2 plus is really low. They don't have a lot. Okay, so which means what will happen is this IP3 will go and what? It activates to open the protein channel and what? Have the calcium 2 plus go back out. Okay, and calcium 2 plus. So the calcium 2 plus will act as what, guys? As a tertiary messenger. Okay, but Is it a tertiary? But they don't call it a tertiary, by the way. They still call this called a secondary messenger. But so IP3 sometimes it acts like what? Open what the channel to have the Ca2 plus and calcium ion to come out, and this will be what? Activating the kinase, the molecules, kinase enzymes, you know, for the next thing that we have to discuss, which is the signal transduction portion of it, okay? But now, calcium 2 plus usually binds to a protein called, if I'm not mistaken, guys, like calmodulin. That's the enzyme usually, it's like calmo, like, you know, Ca, right? That's the enzyme that will bind to a calcium 2 plus, okay, and then activate the kinase, okay? Usually that's, if I'm not mistaken, that's how it works. That's the name of the enzyme, okay, or the protein that will bind to calcium 2 plus, and then activate the kinase for the signal transduction perspective, okay? So again, I'll whenever if I have some time, guys, I'll revisit the tyrosine kinase, you know, receptors and other, like the other guy is really simple, but just take a look, guys. So now the next portion of it, okay. So this is where I think we talked about this last week, okay. The next part is what we call, guys, signal transduction pathway. Okay, signal transduction pathway. So this is where, guys, something that you guys know already, guys, a cascade of the phosphorylation occurs. Okay, and I'm gonna take a little further to the next step, guys, in this. Okay, so because we have to talk about the protein synthesis and stuff like that, guys. So I'll take you to a deeper level today, okay, with the signal transduction pathway. So, because I don't know, like time-wise, I don't know if we can go into DNA replication. I'm not sure, but I think this is like, I don't know, we'll see. So, the cyclic AMP, or like, you know, calcium 2 plus, you know, like activated by the calmodulin, you know, whatever. These guys are what, guys? These are what we call the secondary messenger, okay? So basically what will happen is, guys, the first kinase, so kinase 1, okay? So this guy will be activated by the kinase 1, okay? So once this guy gets activated, okay, with the using of ATP, with the usage of ATP, okay, it will activate the second kinase. So kinase 2 will be activated by the kinase 1, okay? And this will turn into activated kinase 2, phosphorylated. Okay. ATP will turn into ADP and phosphate. The phosphate is now going into the kinase. Okay, that's the definition of kinase, right? Kinase what, guys? Kinase, their job is put the phosphate on the other guy. That's the job of the phosphate, right? And keep in mind, I mean, it's good to know. I mean, who the hell know what's going to be the test, okay? Cyclic AMP, okay? Once they have done their job, okay, you do not want the cyclic AMP to continuously fire the reaction. Okay, if they don't take this out, what happens, guys? They'll continuously fire the reaction, okay? There's an enzyme, okay, where they call the phosphodiesterase. It's good to know, okay? Which will convert the cyclic AMP into just AMP, which is a deactivated form of the AMP, right? So that's so adenylyl cyclase will convert the ATP into cyclic AMP, and phosphodiesterase will convert the CAMP into AMP. Okay. And now the kinase will be turned into activated kinase. We'll turn the kinase 2 into kinase 2 activated phosphorylated kinase 2. Okay, we'll activate the second kinase. Okay. And now once the kinase 2 gets activated, what happens, guys? I mean, it's like, dude, same thing. The next kinase 3 will be activated. Okay, with the assistance of ATP. Okay, the kinase 3 will be activated. Okay. And once this guy has finished their job, the kinase 2 that's activated is what? Like, dude, their job is done, which means what, guys? They have to convert it back to kinase 2, okay? Enzyme that does this is called phosphatase. Okay. Enzyme does this what, guys? Yeah, like kinase, phosphatase. It goes back. Okay. They take the phosphate group out. Okay. This guy, take it out. Okay. Because why? They do not want continuously to fire the reaction by the kinase 2 activated kinase 2. Okay. Once the message is done, they should stop it. Okay. And then what happens, guys? Yeah, so this is what. So next kinase. So it's a whole phosphorylations, okay, of the kinase. But now, like you get like, so you gotta understand, right? Like before, like we move any further down the road, okay? Why they have to like? Okay, so why like not why, but what led them to like investigate all this? Okay, it's like what the hell? Like why do we have to do all this? So the reason behind, if you read the chapter, guys, the reason behind is this. Okay, epinephrine. So the whole reason why like we're doing all like there's a very intricate step. Okay, they're like realizing, oh my god, like there's a lot of stuff going on. Is that what they knew was epinephrine. Like what's epinephrine, guys? Anyone know? Epinephrine, huh?
Adrenaline?
Yeah, exactly. What is that? What does that do to your body?
I mean, I know it activates the fight or flight response, but
Huh? Fight or flight, yeah. So what does that to your body? Like what like more specific to your body, like body cells. What do they do? What happens? Your heart starts beating faster. Like you probably watch. Like I don't know about like you guys, but when I was a little kid, man. I mean, not a little kid, but when I was in high school, like you guys, I mean, there was like a Korean like drama, and then like even in college, like my friend and I were watching and stuff like that, and then like there's a medical drama too, right? And then when someone's like heart stops, like, oh, give me like epinephrine, like like epinephrine. So what happens? Like it makes your heart go, right, beat faster, okay? And also what? It's a fight or flight, just like what he said, right? So heart muscle respond a little differently because what? Like the heart beats faster for them, right? But for liver cells, what happens? Liver cells, they will depolymerase glycogen into glucose, okay? So one of the scientists, I mean, I think the scientist's name is Sutherland. Not the guy name is Sutherland, okay? And basically, okay, so this is what he thought. When he put epinephrine in the liver cell, okay, they start to produce glucose from glycogen, okay? So do you see so there's an enzyme that will break down the glycogen to a glucose, okay, within the cell of the liver. But now, so that what he he obviously, I mean, you don't have to be smart to figure it out, right? Epinephrine activates the cell, okay, inside the cell, which is the enzyme to break down the glycogen to glucose, okay? But now this is now what he did. So in a separate setting, okay, he put an enzyme, okay, that will break down the glycogen to glucose, and he did put the glycogen there, okay? So he put the enzyme, he put the glycogen, okay, and he put epinephrine, okay, like okay, so they have glycogen, it has the enzyme, and he put epinephrine in there, okay. The cell, I mean, so it's not the cell anymore. It's what? Just enzyme and the substrate, okay. And then he put the. So what do you think happened? Nothing. Nothing happened. So nothing happened. So he's like, what the hell? What the hell is going on? Because why? Yeah, now like this is due to the his findings, okay? Because why? Epinephrine never entered the cell. Okay, because what epinephrine is what, guy? Yeah, like it's a peptide hormone, okay? It never comes into a cell, okay? Because why? The enzyme that runs this reaction is what, guys? It's what? It's either a secondary messenger activates the guy, or what? The cascade of the phosphorylation is gonna activate this guy to do this process. So that's a difference here, okay? Like, so he forgot, or he didn't know, there's a lot of stuff going on within the cell, okay? And that's what led to doing like all like, you know, the analysis and the experiments and all led to doing all this, okay? Experiment perspective. So now if you take a look at it, right? So they go into a cascade of phosphorylation like this, okay? And like that, dude, if the last protein is this guy, think about it, if the last protein is that guy, okay, so last protein is what? Okay, this guy, and you phosphorylate this guy, what happens then? Yeah, this guy will start to break down the glycogen, depolymerize the glucose, plasma into glucose, and what? It's gonna work. So that's what this is about. So but that's a response, but again, this process is what we call, you know, cascade of what? Phosphorylation. It's called the cascade of phosphorylation. And this is something that it's something that, you know, is part of what we call signal transduction pathway. Okay. They ask us a lot now. The AP man, they ask us a lot, but Olympiad, maybe like most like one question if they do ask. So, I do want to talk about with you guys because we have to do this like whatever. So, but this is like whatever cascade of phosphorylation. It's nothing fancy. But I think we talked about it, guys. Why they go through like so many kinase of the kinase reaction of the phosphorylation, like, you know, stuff like that. So one of the reasons why we do this is what, guys? For amplification, right? So, you know, one single epinephrine can what? Activate thousands and thousands of what? Of protein, okay? So, which means what? Like thousands and thousands of the protein inside the cytoplasm of the cell can be activated and breaks down the glycogen, okay? It's not only one, it's not a single enzyme gets activated, it's what? Thousands of these guys. So it amplifies the signal, okay? Down the road. So, meaning, guys, I mean, if one single epinephrine can activate what? Like thousands of the cyclic AMPs, which will activate what? Like 10,000s of the kinase 1s, and like it will activate 100,000 of kinase 2. So it amplifies the signal down the road for the cell to what? Make sure that what? All the enzyme gets activated when they get the message, okay? So the purpose, a lot of times, guys, if someone asks you guys, why do they do this? Why do we have a cascade of phosphorylation? Because what? Yeah, amplification of the signal. Okay, that's usually the case for the, you know, the kinase cascade of phosphorylation. But now, what if? Okay, so what I want to talk about now is this. Okay, what if so now we talk about response. Response is where. So what is the response? So a molecule came, okay, so the ligand came and bound to the receptor protein. What becomes the response, okay? So now, if the response is what? Production of a certain protein, okay? So it could be very simple, right? It could be what's very simple as well. Oh yeah, just activate this protein. Just phosphorylate one of the protein and activate that guy, okay? Or even what, guys? Oh yeah, open up the protein channel in the cell membrane, okay? Have some of the, you know, the ion to come in, okay? It could be very easy, right? Or open up a channel, okay? Or it could be channel on the cell membrane to have the glucose come in, for example, okay? So it could be something simple, okay? So it's activation or deactivation, for example, based on the signal they got, right? But now it could be something else. It's such as what, guys? Production of an enzyme, okay? So you're telling the cell, produce this enzyme, okay? As quickly as you can. Okay, that's the signal, right? How does it work? Go to like how do they send the message down to the cell's nucleus to produce this? So listen very carefully now. So the response, I tell you guys, response can be anything, okay? Activation of the enzyme, deactivation enzyme, open up the protein channel, the cell membrane, or it could be what? Like activation of some kind of organelle in the cell. It could be anything, okay? But now, if the response has to be producing an enzyme, then this is the channel we have to take. So this is what it becomes now. So the last protein that gets phosphorylated, okay, last one, let's say K3 is that's the third kinase, last kinase that gets activated. So let's take a look. So let's talk about little lesson today about the protein synthesis, okay? How does that work? So guys, okay? So probably you have a little background of protein synthesis. Okay, a little bit background, guys. I mean, I don't know how much, but we're talking about a eukaryotic cell here, okay, by the way. So, so this is a DNA. Okay, this is a DNA for a eukaryotic cell. Okay, and now on the DNA we have a gene of interest. This is a gene of interest. So keep in mind, guys, the definition of a gene is what? It's a location on a DNA where, okay, it can what? It can produce, or it can code for what? A functioning protein or an RNA. Okay, that's what they call a gene, okay? Functioning protein, okay? So I tell you guys, okay? To produce, I tell you, like something you probably have heard before, like whatever the bio you took, guys, is what? Oh yeah, DNA to an RNA to protein, okay? The very simple process, and that's it, right? I tell you guys, it's a lot more complicated than what you think, okay? So I think the next chapter we'll discuss a little bit more about the DNAs and what form they're in. We talked about, guys, DNA, as we know, is what? In the form of chromosome or chromatin. It's called chromatins, okay? Chromatin is how the DNA is typically found in a cell, okay? And chromatin is what, guys? These DNAs are what? Wrapped around in a histone. Histone is what? Histone is actually is a protein, okay? Where DNA is wrapped around, okay? Keep in mind, guys, histone only applies for eukaryotic cells only, okay? Plus archaea, but I'll put that later, okay? So just to like a little hint of what to be done. Tosh. That's what. It doesn't work.
There's like a powerful on that AC.
Do you feel any cold air?
No.
No?
Wait. Yeah. No, I do. I don't know. I wasn't there before.
Hey, can you yeah. Oh my god, why did they do this? Sorry.
Great, this killed off.
Yeah, it's... It's one of the stupidest things I've ever seen in my life. They'll probably yell at me for doing this.
It's bending a little bit.
Huh?
It's bending a little bit because I can see it, the AC unit now.
Yeah.
Actually, it looks a lot like that in the other room.
Yeah, dude, like other room, I guess someone with the brain actually did it. I can't record this. Oh no. Okay. Okay. No. I was thinking something else, guys. All right. So histone, right? Okay. So for eukaryotic cells, okay? For you, so the keep in mind, guys, okay? The big thing about, so I tell you, like, cell communication is not a big deal, okay? The reason I'm talking about it, guys, because there's more of these questions than the cell communication stuff. So the thing to understand here is, you know, like before we even talk about production of messenger RNA, okay, like producing an RNA from DNA, is regulation. So, like, we can like, dude, talk about like thousands of freaking of these regulations, by the way. So, because like, just let's just think about it, okay? Let's think about, guys. There's an enzyme, okay? Let's say epinephrine, okay? Let's say the epinephrine, if it innervates, I mean not innervates, if the epinephrine gets recepted by the receptor protein of the liver cell, okay? Let's say the response is production of the enzyme that breaks down the glycogen. Let's say which means they have to produce the they have to produce the enzyme. So then you gotta understand, guys, okay? Yeah, what if, like, okay, they have a liver cell, so epinephrine, guys, it'll be all over in the body, everywhere in your body, okay? Because why? Like, it's a hormone, okay? It's a hormone given off by the, you know, adrenal gland, okay? It's like, you know, it's a gland, right? So now, think about it, guys, right? There's a liver cell, okay? There's a skin cell, right? Like, dude, skin cell and liver cell, okay? Yeah. I mean, we haven't really talked about this too much. We didn't take a look at developments and stuff. Liver cell and skin cell, they have exactly the same DNAs. Okay? Yeah. What liver cells have is in what? The skin cells. Yeah, should skin cells break down glycogen too? They don't have any. Okay? So they don't, which means they don't have glycogen. So, which means what? Which means what? Some genes that they have should never be expressed. Like, dude, like, okay, some DNAs you guys have, like dude, my skin cell probably have the enzyme to produce insulin. Should they produce insulin? They should never do, right? Because why? It's not their job to do so, right? So then, if this is a location for production of the protein, okay, then what do they do with this? Which they will never use. What do you think, guys? They will they make sure this portion is what? Never expressed. Okay, how do they do that? And this is where they call, guys, okay, by the way, pre-transcription regulation. Okay, this is like this, a little preview, okay, of the big chapter we'll be discussing, you know, probably a lot of it next week, okay? So it's called pre-transcription regulation. So pre means, because I mean before that, okay, transcription meaning what, guys? Production of a messenger RNA. So from DNA to messenger RNA. And that is what we call, guys, transcription. Okay? Pre means what? They do not even let them produce messenger RNA. Okay, that's called pre-transcription regulation. Okay? So typically how this is being done is this, okay? They methylate. Methylation. So methylation meaning, okay, they have a piece of DNA, okay, on the cytosine, they start to what? They have a lot of these methyl groups on them, CH3 groups. That's called a methyl, okay, CH3. They have a lot of these guys. So, yeah, why do you think, you know, how will this, okay, have the gene never expressed? What do you think? How having the methyl group on the cytosine will have this never get expressed? Why? If a methyl group, so the methyl group is what, guys? Very hydrophobic. Okay, so which means, I mean, it means, dude, like there's an enzyme that needs to get to the gene to get what? To get the gene transcribed. But now, if so many of these non-polar, okay, hydrophobic groups on the DNA near the gene, what happens, guys? I mean, they can't get there. It's all crumbled up. So it's like a piece of paper, right? You have a piece of paper. I want to use my paper. So you got a piece of paper. Phantom of Opera. I don't know what this is. You guys are
That's
doing-
probably important.
Yeah. So, like piece of paper. Okay. So you can read stuff here, right? Like, it's something to read, right? So basically, if you're doing this, what happens is you're doing this. Okay, you're crumbling up. Can you read it now? Can you read it? You can't because what? It's all what? Hydrophobic. Okay, they coagulate. So which means the gene will never surface. You know, the enzymes surface to show show the enzyme. You know what this gene is. So that's one way. Okay. And also, what guys? Deacetylation of histone. So they deacetylate the histones. Okay, what does that do? So acetyl group is what, guys? So they got the oxygen group on it, okay? So the oxygen group is acetyl. So if you have the oxygen group, what happens, guys? It relatively goes to a hydrophilic, but you're deacetylating a histone, which means what? You're removing a polar hydrophilic group from a histone, which means what? We are further making the DNA very hydrophobic, okay? You're crumbling even further. So you got the piece of paper, right? That's it. So you got a piece of paper, right? So you're like doing this, really like doing this, okay? Which means what? Yeah, dude. Yeah, which means what? This will never be expressed, okay? So now, so they make sure that what? The skin cells will never produce what? Insulin. I mean, they have a DNA because every cell has the same DNA. Okay, your skin cell have the same DNA as your your whatever cell. Okay, but now it doesn't get expressed. But now, so that's what they call, guys, a pre-transcription regulation. Okay, they don't even get there. Okay, they don't even attempt. Like impossible to attempt. Okay, but now, if you think about it now, just okay. So let's assume. Okay, this is a very, regulated, portion of a gene. Okay, even this, for the cells, but eukaryotic cells are very tricky, by the way. Okay, they are just it doesn't work. So I'm not gonna go too crazy right now. By the way, just to transcribe a gene of interest for the eukaryotic cells, they need many components, a lot of components. Okay, such as the obvious thing is RNA polymerase. Okay, they need this. They need something, something we call RNA polymerase. Okay, which is what? This is the guy that can produce. This guy is the guy who's gonna run down on DNA and start to produce what? The messenger RNA. Okay, that's the guy. And second guy. So it's the guy called a general transcription factor. Okay, transcription factor. Okay, so and these are assisting, like proteins. Okay, which assists. Okay, the process of transcription. So I'm not gonna go too far with this. Okay, but now keep in mind, guys, the term general, which means what? Every transcription is what? Production of messenger RNA from DNA, right? Every one of those processes, okay, that you guys are aware of is what? This is needed. This is general. This is not like something specific to a certain gene, okay? So these general transcription factors will bind to like promoter zone, stuff like that, blah blah blah, which assist. Okay, that's a whole different day, a whole different game. Okay. But now they need something we call specific transcription factor. Okay, specific, which means there is something that's associated with this gene only. So there is a transcription factor. So, dude, I tell you, the transcription factor is what, guys? I mean, it's a protein that's needed, okay, that assists the RNA polymerase, okay, to what? Yeah, bind to the gene of interest and what start to run down and what produce messenger RNA. So they need these proteins, okay, to form into a complex and what be able to what activate the gene of interest and what start to transcribe it. So they need a lot of components. Not only this, there's more. I'm not just talking about it. So what I'm saying is, okay, the product of the cascade of phosphorylation, okay, perhaps can be what? A specific transcription factor which will what? Activate the what? Transcription process of the DNA, okay? Whatever this is supposed to produce. So let's say this gene is supposed to produce an enzyme, okay, that will break down or depolymerase a glycogen to a glucose, okay? And that's the enzyme that produces, so which means what? The last protein that was phosphorylated from the cascade of phosphorylation and signal transduction pathway is what? Can act as a specific transcription factor, okay? For what? For a transcription complex. So this could be as well be what? The product of what? Product of the signal transduction pathway. So in this case, the response is what? Production of the enzyme. So reception, okay, signal transduction, and response is what? Response is produce the enzyme for the function of whatever, okay? So there's a lot, dude, like we're gonna discuss a lot more of this, by the way. So this is like a very small portion of the whole, like, dude, genetics is a big chapter, by the way. How much of genetics was what, guys? Like 15 to 20% was genetics, the whole test, guys, by the way. And like the cell cycle, he's like 85% at most. Any questions, guys? So like, just give me understanding, guys, like what the cascade of phosphorylation, the final product, can be utilized for. Okay, by the way, okay? Any questions? Any guys? So let me get some water, guys. give me like 2 minutes. Some other receptors, okay, after this chapter. So, so the other, like, just basic stuff, right? Because like more targeting for the AP instead of the Olympiad right now, because again, just like what I said, not a lot of these stuff is not a lot of them is on the Olympiad, by the way. So, one of the stuff, guys, okay, is if the ligand, okay, which is the molecule, the signaling molecule, right? If the ligand is hydrophobic, if this guy is hydrophobic, which is hydrophobic means what, guys? It could be a steroid hormone, okay? What will happen to this guy? So you gotta ask yourself, guys, will the receptor on the cell membrane? So yeah, dude, okay, so there's cell membrane receptors, right? Typically, the cell membrane receptor is what? If the ligand is hydrophilic, okay? Hydrophobic molecule can what? Can come into a cell, okay? Can just what? Enter the cell without, you know, like, yeah, much of an issue, right? Because it's like, dude, this is a lipid, okay? Tiny hydrophilic particles can come in, right? So that which means what? The receptor for these guys are what? Are inside the cytoplasm. The receptors are what, guys? In the cytoplasm. So when these ligands gets bound to the receptor, okay? So keep in mind, guys, okay? When this happens, so typically for you guys, right? For you for the AP, okay? For a lot of times, okay, hydrophobic ligand, okay, binds to the receptor, okay, inside the cytoplasm, okay? A lot of times they do not have a cascade of this phosphorylation. So the kinase phosphorylation, you know, cascade, you know, that usually is happening for the G protein couple receptors, a lot of times, okay? But now the other receptions like this, there's no like kinase phosphorylation, okay? And this can be what, guys? This guy can be just what? Going into a nucleus and what act as what? A specific transcription factors. Okay, so a lot of times, guys, when the molecule is hydrophobic for the ligand, okay, and then receptor is inside the cytoplasm, which means, well, yeah, they might as well just be what? You know, the transcription factor on their own. Any questions? And now, there is, guys, a tyrosine kinase receptor, okay? You don't have to know that for the AP, okay? And I have yet to see a question on that, guys, on the Olympiad, so I don't want to talk about it. So you can read it up on it, guys. I mean, there's a chapter, you can read this. There's a chapter in this, guys, okay, tyrosine kinase receptor. I mean, it's good to read. It kind of reminds me, like, you know, of a little bit of a scaffolding protein, so, you know, but just write it down, guys, like, just scaffolding protein. I mean, that's on the reading probably, scaffolding protein and tyrosine kinase receptors. Okay, so you guys have to do your reading, guys, and you know, just think about the resemblance of those two. You know, when you read the chapter, guys. By the way, okay. But a lot of times, like this is where the two of them that is where the most questions are based on. Okay. Any questions? Let me know, guys. Any questions, sirs? Yeah, I wrote a lot of stuff here about the tyrosine stuff and whatever, but I don't think it's really needed for us right now. Questions? Calcium, like, I don't think there's a need for us to talk about it. Okay. All right, so any questions, let me know. So I think with the time that we have, guys, I think I can finish the cell cycle chapter, by the way. So, because it's not going to take that long. Cell cycle. Let's talk about that. Let's take a look at that chapter. So you might want to read. I mean, it's a very short reading for cell cycle and cell communication for you guys. It's like I don't know, like 10 pages, guys, for both. It's like 20 pages for together. Okay, so make sure you guys read it. It's good to read it, guys, by the way. So this is cell cycle, cell communication. You guys will do this like well into November. Okay, maybe even December. Okay, so yeah, in your school, by the way. So it's like well into there. So. So it's good to read, guys, so that you don't have to, like, you know, really, like, if there's a test, you don't have to spend so much time studying for that, you know, instead of like Olympiad stuff. So, you know, but just so that like learn it now, so that you don't have to waste your time later. Okay, cell cycle. Keep in mind, guys, cell cycle and cell communication only, guys. And there are only like 5 questions combined from like 2004 to 2014. So I have to assign to a different order from this chapter. Okay. Alright, so, but this is something that you need for other chapter as well, so I can't just like skip it, guys, by the way. Cell cycle, okay. This chapter is, so this chapter focuses on mitotic cell cycle, okay, mitosis, okay. So you should understand, guys, cell cycle is, like, to keep it very simple, okay. They have a very simple diagram for this, okay. G1, okay, S, G2, okay, M, C, okay? And that's where the cell cycle starts, okay, and goes around. The cell cycle is. So just to kind of, you know, level set right now, guys, G1 stands for first gap 1 phase, okay, gap 1, okay? The first gap phase of the cell cycle. S stands for synthesis, okay, S stands for synthesis, okay? So what are they synthesizing, guys? DNA. And G2 is second gap phase, okay, gap 2. And M stands for mitosis. And C stands for cytokinesis, okay, division of the nucleus. So keep in mind, guys, I mean, dude, like, you probably have seen this, guys, like, you know, in your like very old AP classes, by the way, AP class, guys. So up until G2, okay, G1 to G2, end of G2, that's called interphase. Interphase, okay? And then mitosis, okay, M, right? They have prophase, metaphase, anaphase, and telophase, okay? So, I mean, you probably heard this, like, right? And that's mitosis. And then C, which is called a cytokinesis, okay? This is a division of a nucleus, okay? So that's what makes them into a cell cycle. Okay, so this is what cell division. That's what this chapter is about. Cell dividing. So if you look at this, okay, so let's take a look at what happens in G1. So G1, guys, is where they call the growth phase. The cell cycle. Cell grows a lot, guys, in G1. So as we talked about this, right? Cell grows. When the cell is really tiny, guys, what happens, guys? Yeah, like the organelles will divide, for example, guys, and so mitochondria will divide on its own, the chloroplast will divide on its own. Okay, cell membrane will get bigger and bigger and bigger. There's more stuff there, so the volume of the cell gets bigger. You know, there's more membrane and stuff like that. So I think we talked about it, guys, by the way. What triggers the cell to divide? So like, okay, so why does the cell divide? Why do they have to divide for whatever reason? So the cell division, okay, occurs because what? For a growth, okay, or for repair, okay, or for more greater efficiency of a cell, okay. That's the main reason for the what? The cell division as we know, okay. Because when they divide for what? The growth and repair, okay, or also what? Like efficiency, as we talked about, right? So we talked about, guys, your simple stuff, the surface area to volume ratio, blah, blah, blah. We talked about that, guys. Okay, that's when the cell triggered to divide. If the number gets too small. So guys, by the way, so when the cells gets too big or they have a need for what? A greater number of cells, for example, then what happens, guys? Yeah, they will go into the S phase of the cell cycle. Okay? So there's a checkpoint here, by the way. Checkpoint, guys, there's a checkpoint. So checkpoint is what, guys? So making sure they're checking what? The cell is normal, the cell is actually okay for the cell to divide. Okay? And we'll talk about the checkpoints, okay? But before that, so like, let's just talk about some generic stuff here. So now, S phase of the cell cycle stands for DNA synthesis, okay? So now, the cells, like, so which means what? Like this, keep in mind, guys, mitotic cell division is what? Make sure, what? A cell replicates and what? Produce exact same, you know, two daughter nuclei, okay? Two of them must be exactly the same as the first parent, okay? That's what we call, guys, what? Mitotic cell division, right? So they have to replicate their DNA, okay? DNA must be replicated, okay? But now, let's take a look at this. Yeah, so you guys are, I don't know, like what, like 16, 17? Yeah, 16, 17, right? You know, sweet 16, right? So just,
yeah,
sweet
I turned 16 this Sunday.
Oh, really? Okay, sweet 16, guys, okay? So, oh my God, I'm old.
I turned 16 last Friday.
Oh, really?
Oh, really? Oh my God, sweet 16, what'd you do? Sweet 16 party?
I went to the city with my friends.
Oh, really? Hey, how is the city like now?
No one wears masks. It's completely like.
Oh, really? I mean, yeah, I've been to a city when my friend was here actually, like over like in June. You know, we were shocked. I mean, how many people don't wear masks?
Yeah.
But I tell you, man, you have to go down to other states. New York like is pretty good wearing a mask. Like, indoor at least they wear the mask, but if you go down to like some states in the south, it's like the mask never existed. They don't wear it at all in any places actually. So that's why like these things are spiking now, I think. So, man, I don't know, but yeah. So okay, so you guys are 16, okay? So long way ahead for you guys. Man, I tell you, man, it's not easy, man. I tell you that for you guys. 16, I mean, the ups and downs, man. But whatever. But okay. So you guys have your cells, and you guys, I mean, I have my cells here, okay? I have whatever cells I have in my body. So some cells, okay, some tissues in your body, okay, they divide more than the others. So where in your body should there be new cells? Like very often, what do you think? So where should they produce more cells? What do you think?
The skin?
Yeah, skin cell, yes. But skin cell is when there's what? A break. Okay, when there's an injury, when they need to repair, for example, yeah, the cells need to divide, right? Well, unless like you guys are still growing, so maybe you guys do a little more. But where else, guys?
Blood cells.
Blood cells, yes. You know, blood cells they have to for example, guys, what was that, guys? I think one of the question or something what I've seen for the cell cycle in the textbook. Okay, because I saw the questions textbook, guys. By the way, before I came to class, guys, and I read the chapter for this. They said the red blood cell will last 120 days. Okay, or something like that. Okay, so which means they have to produce, keep producing, right? So yeah, red blood cells make sense, right? So, red blood cells, all this stuff, they make sense. Stomach cells, okay, so the stomach cell lining, for example, okay, they have to keep them producing. Parietal cells and some other cells in the stomach, they have to keep on producing them as well. Also like hair follicle as well. I mean, they have to, they have to also keep on dividing and producing, mitotic cell division as well. But what about the cells that never divide? Is there a cell in your body that never really divide at all, ever? What do you think? Anyone? What do you think? Cell will never divide at all. Absolutely never.
Like since you're born?
Yeah, since you're born. Some cells will never divide. Which cell are those? So you gotta think of it like this, right? Can you, like, if you guys think about it, right? Can someone replace, a personnel who's flipping burgers in Burger King? In McDonald's, Burger King, can you hire someone like quit, like out of blue, right? And that guy's flipping burgers, flip burgers, like, Can they find a replacement like quickly on the day? What do you think? Yes? Yeah, I think yes. Just bring someone in. I'll pay you like $20, man, an hour. You want to flip burgers? Dude, I will do it, man. But it doesn't require a lot of expertise. Like, yeah, it's not that hard. But now, if you think about it, right? If you are like a CEO of a company, okay, or like for example, you're like like Joe Biden. Like, can you replace Joe Biden like that, and then it's gonna function like as how it was normal? Like Joe Biden, it's just gone out of blue. You wanna do it? Like, I'll pay you $20. Does it work, guys? You can't replace something like that. So what I'm saying is, high functioning cells, very complicated high functioning cells, they can't just replace it in 2 days. Okay? Well, day, right? Because they're not flipping burgers. So what I'm saying is, some cells in our body are very technical, very complex, okay, and they have a very specific job. Such as neurons, nerve cells, very specific. They have a very complicated workflow that they have, okay? Which means what? Yeah, like nerve cells will never divide. So you probably heard, right? Like someone got on a motorcycle, they got into an accident, they got injured from the lower back down or something, and then what happens? They never get to walk again. Okay, because why? I mean, dude, yeah, think about it, guys. If you get a cut of the skin, okay, what happens, guys? Oh yeah, it grows back, there's no problem there, right? If someone injures their nerve in the back, what happens? They never go back as well. I mean, you probably heard like a miracle occurred, and sometimes some people can walk, but never. 99% guys, they're gone. Because what? Nerve cells will never go into the S phase. They are stuck at something we call G0 phase, by the way, just so you know. So a muscle cell as well. Think about it, guys. If you guys go start working out, like like the Olympics going on right now, like these people that lift really heavy weights. If you work out, then you get big, okay? Do you think the muscle cells are going into mitotic cell division? Absolutely not. Just the muscle cell that you had are just getting bigger, okay? So the muscle cells, okay, and the nerve cells, the type of cells that is what? Like those are Joe Biden's, okay? They never get to divide, okay? They're just what? Yeah, like if you dude, you've probably seen if someone had to cut their muscle out, it never grows back. So it never grows back. So which means, guys, those are the cells where they're very specific and complex. We're just stuck at what? G0 phase. It never grows. Okay? You have to get transplanted for the muscle cell from one thigh to the other, but it never grows back. So, and those are the cells, but skin cell, man, you lose it every day, but those are what? Like flipping burgers cells, So just so keep in mind, guys, just so you know. But once they're about to divide, right here, okay? So you guys are 16, I'm 21, so but so keep in mind, guys, okay? Would your cell, so the cells that divide a lot, right? Your stomach cell versus my stomach cell, are there any difference? What do you think? And how many times can it divide? What do you think? How many times a cell can divide? What do you think? A normal cell, how many times can it divide? How many times they're gonna do this in your lifetime? What do you think? What do you think?
Countless.
Countless, million times?
Yeah.
What about you?
Like more than a million.
More than a million. Oh my god. 50. That's it. around 50, maybe 200 guys. That's it. Gosh, yeah. So, like people, like dude, like just so you know. Keep in mind, guys, like human, we cannot like, dude. You guys watch too many movies, guys. By the way, it's like some students. We're not like, we die because what? These cells or what? You can't divide anymore. Okay, so they divide and divide. They kill the old cells and stuff like that, right? The reason, so that what I'm saying is, S phase of the cell cycle represents what we call the DNA replication process. Okay, so the DNA replication, okay, there is a limitation of how many times you can divide. Okay, but now once it reaches that point, the cell is gonna kill themselves. By the way, so it could be any part of the cell body, guys. So it will not. So you probably hear like some of the old people like, oh yeah, if I get a cut on my arm, it takes a really long time to heal. By the way, because what? Like yeah, the cells are done. Like I'm checking out. Okay, so because why? They're too old, so they can't divide any further. So the reason is, keep in mind, guys, this is something we're going to discuss next week probably because the cell DNA replication process. So keep in mind, so the DNA, this is like basically the DNA, double-stranded, right? At the end of it, so the important genes of the DNA are like here. Okay, so it's in the inner portion of it. Okay, at the end, they have something that it's never transcribed. They don't use. Keep in mind, they don't use every portion of a DNA. Okay, at the end, they got something called telomeres. Okay, they got something called telomeres. Okay, so these telomeres, they never use. So they never use that part. They're there for a purpose. The purpose is every time a DNA replicates, they are gonna get a little shorter and shorter and shorter. Okay, the DNAs will get shorter. So DNA, if this is, for example, guys, like 1,000 kilobases, okay, for one each. Keep in mind, once the DNA replicates, guys, during the S phase of cell cycle, it will not be 100, guys. It'll be what? A little shorter, 97. Okay, so and then if it divides one more, 90, 90, 92. Okay, so they will get progressively as they divide, the DNA will get shorter and shorter and shorter. Okay, so why is something to discuss next week. Okay, but now once if they divide enough, what happens, guys? So once the telomere is all gone, what happens, guys? Then yeah, the cell will start to reach what? Where they have the genes. Your cells will see this, will notify, and will be what? Aware of this, and what? It kills the cell. Okay, once the telomeres get too short, what happens, guys? The cell's gonna, oh yeah, bye-bye. I'm gonna what? Kill the cell, terminate the cell. So that's the reason why your cell, we are on the clock, okay? So your cell versus my cell, if we compare our telomeres, your telomere is longer than my telomere, okay? So, because you guys are younger. Okay, your cells divided less than me, okay? So that's one of the ways to kind of tell the age of a person, like how long the telomere is. If you're really old, like it might be a little, like depends on DNA as well. But you have to ask yourself, guys. There's some cells can divide more than 50 times, such as like in a sperm cell, for example. Like in a sperm, someone can produce sperm until they die. Okay, the male can produce because why? They got something called telomerase. They have an enzyme called telomerase, which can extend the telomeres, and they can divide as many as they want. Because why? Yeah, they got telomerase, which can what? Yeah, dude. Like, okay, they extend the telomeres. Okay, how nice. Okay. Also, cancer cells have telomeres as well. Okay. So, one of the what do they say? One of the cancer treatment is what? Make sure what? Like one of them is what? Like there's a lot of cancer treatment, by the way. There's many cancer treatment, guys, by the way. One of them is what, guys? Deactivate telomerase. That's one of the cancer treatment, by the way. Okay. So I mean, again, like there's a lot more we'll discuss, guys. By the way, so S phase what? Yeah, replicate the DNA. Make sure that what? You get an equal copy gets to the what? To the daughter nuclei. Okay. And once that's done, it goes to G2 phase. G2, guys, by the way, is the they call this a second growth phase. Okay, so they're getting themselves ready for cell division, by the way. So the second G2 phase, by the way. Okay. And then there's a checkpoint at the end of G2. Make sure that things are ready, guys. We'll talk about the G2 point checkpoint as well. And now, so the M phase. So this is like M phase, guys, mitosis phase. Nothing fancy other than what you should already know, okay? You should know, guys, there's a prophase, there's a metaphase, there's an anaphase, there's a telophase, okay? Nothing fancy, okay? Like, I don't even want to draw this right now, guys, by the way. So it's whatever's in the reading, guys, are very, very self-explanatory on this one. And you will not have a question on this one on Olympiad. Like, they won't ever ask to come out and say, explain about my talk. It's too easy, okay? It's like freaking 7th grade stuff, okay? They don't have questions on this. So, first and first, guys, during the prophase, I'm going to be very quick on this, okay? Prophase, guys. So first, we're talking about the M phase here, okay? The M cycle, phasal cycle. So mitosis. So the prophase, basically what will happen is, basically what will happen is, the DNA is now what, guys? The DNA and histone, the form is a chromatin. So they use a term, something called condensation, okay? So what will happen is DNA and chromosome, okay? That means DNA, which is in the form of chromatin, it will condense into chromosome. So chromatin will condense into chromosome. Okay. Chromatin to chromosome. Okay, so chromosome, you probably know what it is, guys, by the way. Chromosome is what, guys? So chromosome, you've probably seen this, by the way, so this is what it looks like. Okay. So keep in mind, guys, it has 2 sister chromatids. Okay, so it's got 2 chromatids, okay, 1 centromere, okay, and now these are the protein called kinetochore. So keep in mind, guys, by the way, so in order to be a functioning chromosome, okay, you need to have 2 sister chromatids, and it doesn't always have to be 2, sister chromatids, and also what? Chromatids, centromere, and what? Kinetochore. Okay, so that's basically what we can kind of say as what a chromosome right now, okay? So keep in mind, guys, these 2 sister chromatids are exact replica of each other. So when a cell goes into a DNA replication, they get what? This was what it was before, and what? They made a copy of it, okay? That's what's happening. And now, the whole process of replication, guys, that's something to discuss, okay? But now, like, tangled up chromatin is what? Turned into what? A nicely condensed form of the chromosome. That's what happens in the prophase. And also, by the way, the nuclear membrane disintegrates, okay? The nuclear membrane goes away. And also, nucleolus goes away, okay? You can't really see the nucleolus anymore. And also, they start to produce, okay? They call it spindle fiber. Spindle fiber. So these are what, guys? These are the microtubules, okay, which will be created in the centrosome, which will be what? Assisting of the cell division. So we talked, yeah, do you remember, guys? Microtubules, right? Remember, guys, what I said when we discussed microtubules? So microtubule is what? Microtubule is, this basically cytoskeleton, which is made up of the tubulin protein, right? We talked about this, okay? So there's a microfilament, microtubules, like intermediate filaments, by the way. So now, microtubule, I mentioned this when we discussed this, okay, is what? This will be what? Assisting in what? The mitotic cell division, okay? So now, you should know, because the microtubules, it gets produced in what? Centrosome, okay, centrosome, right? Centrosome is where they have a centriole for the animal cells, and what? There typically is where they start to produce microtubules, right? And now, they have a name for the spindle fibers, a name they put a lot, for the cell cycle microtubule, okay? So they start to produce spindle fiber, and typically their job is what? To make sure, that these chromosomes line up properly for the cell division, okay? So they start to produce microtubules for the spindle fiber chromatin condenses into chromosome. Okay, now also what, guys? Like the nuclear membrane goes away, nucleus goes away, blah blah blah. Okay. And now they got the next stage. What, guys? They have something we call the metaphase. Okay, so metaphase what? I mean, again, so these chromosomes, they line up in the midplate. Okay, so. So guys, by the way, so this is what happens, okay? We got chromosomes here, okay? And then what happens, guys, they line up in the midplate. So they're all tied to, you know, these microtubules. And then this is the metaphase plate, okay? And keep in mind, guys, by the way, so microtubules are attached to the connectochore, okay, of the chromosome. Okay, so again, the connectochore is the protein that's on the centromere, okay, of the centromere, which is centromere is the one that what connects the two, like hold on to the two chromatids. Okay, connectochore, their job is what? Not their job, but yeah, their job is to what? Yeah, make sure that they're hooked onto nicely to the microtubules. Okay. That's what we call the metaphase. Okay, metaphase is what? Yeah, all the chromosomes line up. And then during the anaphase, that's what they call, guys, anaphase. Make sure you do the chapter, by the way, okay? Anaphase. So anaphase is where, okay, you guys should know, like these two chromatids are gonna separate, okay? It's good to know the enzyme. So there is a protein inside centromere called cohesin. Cohesin is the protein that holds down the two chromatids together. Okay, they're being held down. Okay, and now cohesin is being broken down by the enzyme called separase. Okay, so the enzyme separase. It's a very simple name, guys. Enzyme called separase, okay, which will break the cohesin protein and what? Separate the sister chromatid, break them down, okay? And then what happens then? And then what will happen is the broken chromosome now into what? The sister chromatids are gonna start to move down to one side. So now these guys are gonna move down to what? One pole, you know, of the cell. So now there's a talk about this, right? So yeah, listen very carefully. Yeah, we talked about this, right? How do they go from here to here? Like, how do they move from here down to this? What do you think? So we remember we talked about it, guys. Inside the cell, right? The vesicles have motor proteins, and what? They use the microtubules as a rail, like a rail they follow. That we talked about, right? So very same thing. So in this case, motor protein is like connectochore, acts like a motor protein, okay? And they follow the microtubules and to run down to what? To the other side. So it's like, well, I mean, will this feel Olympiad? I don't know, man. But we'll look at AP now. AP, they think this is too much, okay? So the connectochore acts as a motor protein and what? Okay, it will run down the microtubules and what? Will head down to the one side of the pole. Okay, that's what happens, guys, during the anaphase. Okay. So cohesin, separase, okay, connectochore acting as a motor protein will run down the microtubules, okay, to the other side of the cell. Okay. Before I do anything right now, okay, so very simple, you know, question. And some students get it wrong. How many chromosomes do you have in your cell, Derek?
23.
23.
Oh wait, no. Isn't it the other way? Double 46.
46. You got 46 chromosomes, right?
I know.
I got 46, hopefully. I mean, hopefully, like, you know, if I go, dude, I don't know, but so let's see. Let's take a look at this, okay? So, yeah. So let's take a look at this. 1, 2, 3, 4, right? If you look at this, this is what it looks like right now. Okay, so I'm just gonna draw this, right? So like human, right? We say our what? Like we have 46, but we write it like this: 2n is 46. That's how we write this. 2n is 46. Okay. And now, is the reason why we write 2n is what? Because our number of chromosome is a diploid, okay? Which means like we got one set, okay? In that one set, we got what? 2 of homologous chromosome. I don't want to go to creation right now. Okay, homologous chromosomes. Okay, but okay, so like not but like okay, so how many chromosomes does this thing have right now? Okay, so 1, 2, 3, 4, 5, 6, 7, 8, right? Let's go 8. Look at 8, right? 8, right? So let's say we have that many. Okay, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7, 8. Let's say that's how much we have. Okay, 8 chromosomes. Okay, and okay, so now you gotta be very careful now. Okay. They are now separated. Okay, so this is they haven't really got divided yet. Okay. 1, 2, 3. So 1, 2, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8. Okay. So now they're separated. Okay. Do you see that, guys? So how many chromosomes do we have now? How many chromosomes is this? So okay, so yeah, how many chromosomes is this, Derek?
The same amount?
So is it still 1, 2, 3, 4, 5, 6, 7, 8? Is that what it is?
Yeah.
So this is very tricky, okay? So now. So right now, I mean, so like even like freaking like whatever, like don't even look at this. So basically, so before the DNA replicates, this is basically how they should look. Okay, the chromosomes. That's how they should look. Okay. So which means what? They're not paired. There's no sister chromatids. So creation of the sister chromatids occur what? I mean, in terms of chromosome perspective, I mean, it's like let's say the DNA is all chromosome. Okay. So sister chromatid gets produced during the S phase, okay? So that which means like during the G1 phase, that's what it looks like, okay? Now how many chromosomes do I have? How many chromosomes is this? 1, 2, 3, 4, 5, 6, 7, 8. This is 8 chromosomes, okay? But now, if they pass the S phase, now they're in what? G2. What happens then? Okay, they have what? Now they have sister chromatids. It's replicated now. Okay, so now they look like the normal chromosomes that you guys are aware of, right? How many do I have? How many chromosomes do I have? Still 1, 2, 3, 4, 5, 6, 7, 8. So this is very tricky, guys. Number of chromosomes, it's just like, you know, it's what it looks at. Wow, there's a whole one thing. That's a chromosome. So which means like if you have a chromosome that looks like this versus what, just one, like just one chromatid, this is one chromosome that's one chromosome as well. So number of chromosomes is what? The same. Okay, like number. So what I'm saying is, the number of chromosome doesn't really tell you about what? The amount of DNA for the cell, okay? But now, this one is what? Yeah, it has one chromosome, but it's what? It has 2 chromatids, okay? So, take a look at this. Going back to this. So, it initially had 8 chromosomes, okay? But now, it's like, you know, like before the cell has been separated, what happens, guys? This is 1 chromosome, 2, 3, 4, 5, 6, 7, 8. 8 chromosomes here. Well, here, 8 chromosomes on this side, which is what? Total right now is what? You got 16 chromosomes overall right now. So all they did is what? The cohesion is separated by separase, and now, yeah, they're trying to walk towards the other side, okay, using the motor proteins. You know what happens, guys? But now, yeah, now you really haven't added or replicated more protons, or I'm sorry, replicated more DNAs. But what happens, guys? Number of chromosomes doubled. Okay, so now how many DNAs? Like, yeah, you fucking have double the chromosomes now. Okay, so the number of chromosomes doubled. Okay, you know, because like what happens? Like, yeah, because all they did is well, yeah, just separate the two. But now amount of DNA hasn't really changed during the mitosis, but still amount of chromosome did change. Okay. So that's basically what we call the anaphase, okay? They're starting to separate. And the telophase, okay? The next one is called the telophase or telophase, whatever we call this. Telophase is just the opposite. It's the opposite of the prophase, okay? So, which means, guys, I mean, as they move down to one side of the pole, they start to the chromosome starts to dissolve back into chromosome. They start to produce the nuclear envelope. They start to, you can start to see nucleolus again. you know, stuff like that, okay? And then it gets ready for what? The cytoplasmic division, okay? The cytokinases. So now, do you guys recall? Okay, do you guys recall, guys? There's something about the cytoplasmic division, which is assisted by one of the cytoskeleton. What was it?
The actin filaments?
Exactly. There's a what? There's a
Cleavage for-
microfilament. Okay, that what helps them to break or to what? To break the or to separate the cytoplasm. Okay, that's called what? It's called the microfilament. Okay, it's cleavage furrow. That's what it is. Okay, so it's like it's like you're tying your shoes. Okay, like tightly. Okay, and that's what that assists to separate the cytoplasm in half. Okay, it doesn't separate like that, but and then that's what that becomes, guys. You know, the C phase of cell cycle where it's cytokinesis or division of what? Division of the cytoplasm. Okay. It's dude, this is like nothing fancy. Like it can't get harder than this. Okay, like what is there to ask here? The one thing to ask here is, you know, where the checkpoints. Okay. So on the AP, like you guys were talking about the checkpoints. So there are 3 checkpoints you guys have to know. Typical 3 checkpoints: G1, S, G2, M phase, cytokinesis. Okay, there are 3 checkpoints. One at the end of G1, before it gets to S, at the end of G2, and what during the M phase. So there are 3 checkpoints. Checkpoint 1 to 3. Okay, after G1, G2, and M phase. Okay. So how do they pass the checkpoint? Okay, how do they know? Okay, so yeah, time to go, and time to go past the checkpoint. Okay, how do they do that? And what makes them not pass the checkpoint? Okay, and stuck at G0 phase. So there are something you have to know. Something we call cyclin. Okay, so they increase the amount of a molecule called cyclin. Okay, and there's something they call cyclin dependent kinase, CDK. It's called cyclin dependent kinase. So yeah, dude, like kinase again, like dude, the kinase is something talked about a lot in bio. Okay, so you know, so which means what? CDK. So which means if the cell is supposed to go past the G1 phase. Okay, so keep in mind there are different cyclin, different CDKs, by the way. Okay, so which means that they have a different CDK cycling, guys, on all 3 checkpoints. They're not the same thing. So if they want to go past, if the cell is allowing to go past the G1, okay, then what happens? There's a fluctuation of the cyclin amount, and what happens, guys? Cyclin amount will go up. The cyclin will bind to the CDK, okay, and CDK, of course, what? Will phosphorylate something that will trigger the cell to go into the S phase, okay? So basically, what cyclin and CDK, once they combine. Once they combine, guys, they call this MPF, I think. MPF. Okay. If I'm mistaken, it's a maturation promoting factor or something. I don't know. That's you can find that in the reading, by the way. Okay. So, which means what? This is what letting the cell to go to the next phase. Okay. Without the cyclin and CDK, what happens, guys? Yeah, the cell's not going to go. Okay. So there's one interesting thing that you can read, by the way. So, you know, so for example, guys, if you put, for example, like, dude, I'm not kidding. If you put a nerve, I mean, so I mean, like, dude, this becomes like the next scientist, like, thing you guys can do. Okay. There is a saying, okay, where, you know, if a cell is currently going under mitosis, okay, there's a cell going into mitosis, okay, which means what? There is a CDK, okay, there must be CDK in what? And CDK and cyclin level must be at a certain level, okay, in order for the cell to go to the M phase, right? So, but now the thing is, so this cell is currently in the M phase, and now you added one cell that is what? Stuck in G1, okay? You fuse those 2 cells together, which means like, okay, you fuse the 2 together, okay? Like, you know, like gap junction, blah, 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 right? Yeah. So what the hell is gonna happen? So one cell is G1, okay, stuck at G1, okay, the other is M phase. What do you think? So what will happen is the G1 the cell that's in G1 will immediately go into the end phase. So it will what? It will go into the end phase. It will start to go into prophase and metaphase and anaphase. Okay, the chromatin will start to condense into the chromosome. Okay, they will start to. So like, what is there to divide? And cell hasn't replicated, but the DNA hasn't replicated. But what? It goes directly to the end phase. So it tells you what? Okay, there's what? Think about it. Yeah, whatever that's in the cytoplasm of the end phase cell is what going into this and trigger the cell to go into what? Go into the end phase. Okay. But now this is where, like, the future scientists are, I'm too old, man. I mean, I'm not 21, so if you figure I'm not 21, yeah. So you got to think about it, guys. Okay, what if you are in a lab, what are you gonna do? Yeah, what do you think you're gonna do? Like, okay, like you heard this right now. What are you gonna do? If I were you guys, if I'm in a lab, I'm gonna put this guy, which is a muscle cell or a nerve cell, maybe fuse that guy to this guy, see if this replicates. Do you know what I'm saying? Yeah, like that's what I would do. And then what? If this guy replicates, then what happens? Oh my God, I'm a millionaire. Because yeah, you can cure the guy. It's like I don't know. So that's one of the things. I mean, not only studying for these garbage stuff, by the way. So you guys have to apply some of the stuff you're learning, and like, oh my God, when I go to college, oh my God, what if I put this there? Then what's gonna happen? That's something to think about. So yeah. So but because what? Like if you put this cell, this G1 cell, into the G2 cell, for example, like I don't know, like, and then like combine these two together and then add a cyclin CDK, that's gonna pass the 2-second checkpoint. Yeah, it's out within phase. If this hasn't really gone into the DNA replication, it'll still go because of what the signaling is what all happens based on cyclin and CDK amount. Okay, so as something to note, okay, something like, well, I don't know, like if you guys like bio, because I tell you guys, bio is not my favorite. Bio, I think, is garbage. I don't say garbage, by the way. At least the Olympiad is. Okay, Olympiad it's a hard test. Okay, it's obnoxiously hard. Okay, that's one of the reasons why. But like, just not just for building your resume. Like, think about stuff like this. Okay, like, okay, oh my God, what about this? What about that? It's a good thing to know. Okay, I don't have the answer, by the way, because I'm not studying these stuff. Okay, by the way, I'm not into these things. But I'm something to that. Okay, it's something interesting. Okay, all right, guys. I'll assign you the homework, but it will not be Olympic questions, just so you know, because I'll assign you. I mean, there are like 4 questions on them. I have to assign something else. Okay? All right, guys. I'll talk to you guys next week.